It's the most complete lineup anywhere on the planet. Join us for another progress report. Let's talk gene modulation. I'm here with Kirsty McLean, our application scientist, our specialist in this area, talking about gene modulation. What kind of exciting things do we have here at the show?
We're at the AACR and we've got tools for gene modulation. We're going to be spotlighting our siRNAs for transient knockdown effects, our shRNAs for creating stable cell lines. Perhaps you've got difficult to transfect cells, or perhaps you want to work in vivo. There's a lot of mouse models out there that people want to use for translational work, to modulate human diseases. Of course, that can come as single targets, or perhaps you're interested in the entire genome. So we have over 700 human and mouse microRNAs. So we have options for that. Of course, we've got our shRNA options. Maybe you've got a difficult to transfect cell type, or maybe you want to work in vivo. You want to create some of these mouse models that can mimic some of that human disease. Of course, we can overexpress your genes of interest, by the use of cDNAs and ORFs, and of course they're very amenable to cloning. You can add your reporter tags and you can do gene localization, so it's not just about some of those RNAi rescue approaches that you've heard about. But while we're at the cancer meeting, let's not forget about microRNAs, which have obviously become very important as far as cancer research, because clearly their deregulation contributes to some of that metastatic and cancer phenotypes, that transformation event that occurs. So of course we've got tools to address some of those microRNAs. We have microRNA mimics and we have microRNA inhibitors, and that's our hairpin inhibitor design that we came out with a couple of years ago. And of course, perhaps you don't even know what microRNAs are being expressed in your given cell type or tissue, so we also have profiling services available for that. Now, of course, we've got the entire genome covered as far as microRNAs, and clearly that's up to date for the Sanger database. But while we're here at the AACR, we're going to be launching what we call our vector-based microRNAs. These are termed mimics. And again, that affords the use in these difficult to transfect cell types. And again, perhaps you want to work in vivo and create some of these mouse models. And certainly that can help address some of these issues, these long-standing issues associated with cancer research and perhaps designing novel chemotherapeutics.
Well, we try to keep it short, but there's so much more at thermoscientific.com.